Welcome to the Advanced Fluorescence Microscopy and Image Processing course. So today is the 7th lecture and we will discuss about types of illumination including epifluorescence, TIRF, that is total internal reflection fluorescence, HILO, that is highly inclined and laminated optical sheet illumination, then the light sheet illumination and two photon illumination. So let me first explain that why this different kind of illumination is required. So as we discussed in the last class, so that generally in the epifluorescence illumination or the wide field illumination, what happens, let's say your objective lens illuminate the entire cell, okay. However, it collect the information only from the focal plane, okay. So what is our loss here? So what we are losing here? So the entire cell is getting illuminated. That means we are exciting all the fluorescent molecules. So they will start photo bleaching and they will create phototoxicity to the cell. However, we are collecting information only from the focal plane. So that is our loss. So we illuminate the entire cell, but image only a fraction of it. And this causes unnecessary photo bleaching and photo damage. And when you image this focal plane, you will get the out of focus fluorescence light from these molecules, which are above or below the focal plane. So these molecules are also illuminated. So they gives fluorescence, and that fluorescence is known as out of focus fluorescence, and that will increase the noise in your image. Okay. And what are the advantages of this epifluorescence? So it is most easy and convenient to set up. Most of the microscope works under this epi illumination mode. So this illumination mode is useful for a quick glance at the sample. So let's say you have transfected some cells and you just want to check that whether the GFP protein has expressed or not. So you can immediately go to the epifluorescence microscope and check that whether the GFP expression is there or not. And it is also cheap compared to the other illumination strategies that we will study today, and no specialized optical components are needed for this kind of a epifluorescence microscope. So it is most simplest form of illumination with some disadvantages, but for a quick glance of the sample or for some basic applications, it is still a good option. But let me show you, there are some advanced options for illumination to avoid the unnecessary photo bleaching and photo damage. So these are the different types of illumination. So you can see this is your slide and this is your cell. So in the epifluorescence or the epi illumination mode, you see that the entire cell is getting illuminated. However, the focal plane is this. So your objective lens will only collect the fluorescent signal from this focal plane. So even though the other molecules which are present above and below the focal plane, they are illuminated, but you cannot capture the fluorescence from those molecules. So in the confocal illumination, confocal microscopy uses laser-based illumination, and because of that, you can focus the light at one particular spot on your sample. So here, if you see in confocal, this kind of cone of the cone-shaped area is illuminated, not the entire cell. Then the other type of illumination is the TIRF illumination, that is known as total internal reflection fluorescence. So here you see the light is passed from this particular angle. We will discuss more about this in the next slides. But light is passed, the incident light is passed with this angle so that it will completely get reflected from the surface. And because of this reflection of the light, it produces the evanescent field, which is perpendicular to this direction. And this evanescent field will illuminate the fluorescent protein molecules which are present in your sample, but it can only illuminate up to 100 or 200 nanometer in depth. So let's say in this cell, only the area which is just attached to the glass surface or the slide or the cover slip, that area will get illuminated, but you cannot illuminate the fluorescent molecules which are either present in the nucleus of the cell or even on the other side or other side of the cell. The other type of illumination is the light sheet illumination. So here you can see a thin sheet of light is passed from this side and it passes through the sample. So thin sheet of light illuminate the sample. Okay, so this way you can avoid the fluorescence from the molecules which are above and below the focal plane. So this is your focal plane, and you are passing a sheet of light at the focal plane. So this way you are avoiding the illumination of the molecules which are above or below the focal plane. Then this is a two photon illumination. So by two photon illumination, you can particularly illuminate one point kind of a structure in the cell. Okay, so the entire cell is not illuminated, only this one point kind of area is illuminated. And in the high-low illumination, you can see a thin sheet of light passes through the sample. So the light is entered into the objective lens at certain angle, so that a thin sheet of light pass through the sample. It can pass through the nucleus as well as in the cytoplasm. It will only illuminate a few molecules present in the nucleus and in the cytoplasm. So these are six major types of illumination strategies, and now in the next slides and throughout this lecture we will discuss in detail about each of these types, that how this kind of illumination is achieved, what are its advantages, disadvantages, and in which particular case you should use this kind of illumination. So let us start with the first one, that is turf illumination. So we have a separate lecture on the confocal microscopy. So today we will not discuss more about confocal microscopy, but today we'll start from turf, high-low illumination, light sheet, and two photon illumination. So let us start with the turf illumination. So TURF stands for Total Internal Reflection Fluorescence Microscopy. So sometimes it is known as TURF-M, or sometimes it is known as TURF, Total Internal Reflection Fluorescence. So TURF-M exploits the unique properties of an induced evanescent wave or the field in a limited region immediately adjacent to the interface between two media, that is cover slip and sample, having different refractive indices. And why turf illumination is important? Because it reduces the background fluorescence from the out of focus fluorophores. And this is a best method for studying cell membrane or wall associated biomolecules and for in vitro single molecule assays. And this technique is not good for the 3D optical sectioning of a sample. Okay, so in the last class we discussed about the 3D imaging, in which you image the entire 3D volume of the cell. So this with TURF-M you cannot do the Z stacking. Okay, now how this uh, uh, technique works. So here you see that the incident light enters at particular angle, and it is entered in this angle is set in such a way so that the incident light is completely get reflected. So this is the incident light and it is completely getting reflected. Okay, and because of this reflection it produces the evanescent field. Okay, so this is a physical phenomenon. So when light completely reflects from this surface, it produces the evanescent field at the interface, and that evanescent field decay exponentially in this direction. So what does it mean? Let's say this field can illuminate the molecules which are very close proximity to this interface. Okay, so just about 100 to 200 nanometer distance, the evanescent wave can illuminate the fluorescent molecules. So we'll look into the details. So see, here I have shown the difference between epifluorescence and turf fluorescence. So see, epifluorescence illuminate the entire cell, and because of you can see there are so many fluorescent molecules inside the cell and all of them get excited at the same time. Okay, even though the focal plane is here and you can collect the fluorescence from only one focal plane, not from the entire cell. Whereas in turf microscopy, you see this is the incident light. Incident light enters at particular angle, and that incident angle should be greater than the critical angle for turf. So what is the critical angle? So the critical angle is the angle above which the light shows total internal reflection. So see here, the theta c is the critical angle, and the angle of the incident light is slightly higher than the critical angle. So whatever the light enters it will get reflected from the surface and it will produce the evanescent field. Now see, this evanescent field is only for 100 to 200 nanometer. So you can see in this cell, not all the molecules gives fluorescence. Only the molecules which are in very close proximity to the surface or the interface, they can only give fluorescence. And because of that, this technique is really useful for studying the membrane bound or membrane associated processes. Let's say if you want to see something in the cell nucleus, you should not use TUF, because the nucleus is much inside the cell, and even this by TUF illumination you cannot illuminate the molecules which are present in the nucleus. So this method is good for studying the membrane associated processes or membrane bound proteins, but not good for studying anything which is intracellular or even which takes place in the nucleus or even in the deeper area of the cell. Okay, so below you can see two images for the same cell. This image is taken by the epifluorescence mode and this image is taken by the TUF mode. So in the epifluorescence mode, you can see there is a lot of stray light. Okay, so that stray light is nothing but the fluorescence from the out of focus molecule. So here you see all the molecules are getting excited. They are giving fluorescence. Okay, but this objective will collect the fluorescence only from one particular focal plane. So what will happen? The fluorescence from the molecules which are above and below the focal plane will increase noise in this image, and that's why you can see that here you cannot see the individual molecules clearly because of the lot of noise from the out of focus molecules. Whereas here in the TUF illumination, only the membrane associated molecules are excited. So all these other molecules, they are not illuminated. Okay, and because of that, there is no out of focus light, and because of that, you can see the very good signal to noise ratio, and you can clearly see the individual molecules. So let me explain how this TUF illumination is achieved. So here you can see the light path for TUF M illumination, and so generally in the optical or in the epifluorescence mode, you can see we use the LED or the other metal halide or mercury arc lamp as a light source, and it illuminates the sample like this. But in TUF, generally TUF is laser based, and laser produces the coherent and collimated source of light. So that light source is collimated. So you can see a single thin line is passing through the objective lens. Okay, and just by changing the angle of this mirror, you can change the angle of the incident light. So you will change the angle so that the light ray will pass like this, and it will bend on this side. The more you change the angle, you can see the light will bend more. At this particular position, you can see the light is almost now parallel to the uh, uh, your sample plane, okay, or your slide or the cover slip. And even if you go further this way, then you can see the total internal reflection of the light ray within the objective lens. Okay, so whatever the light entering, it is completely getting reflected and it is coming out from the other side. Okay, and this total internal reflection of the light develops evanescent field here, which can illuminate the fluorescent molecules which are in close proximity within 100 to 200 nanometer. So this is how the TUF illumination is achieved, and this is known as objective based TUF. So let me tell you that this TUF illumination can be achieved by two ways. One is known as the prism based TUF, and other one is the objective based TUF. Most of the advanced microscopy technique use the objective based TUF, but there are ways by which you can use the prism based TUF also. But for the prism based stuff, the alignment is little tricky and difficult to reproduce. It is much cheaper, whereas this objective based stuff is little expensive, but it works better with the high magnification and high numerical aperture objective lens. So in most of the advanced microscopy techniques, generally objective based stuff is preferred. And what we saw here, this I have shown the objective based total internal reflection instead of a prism based total internal reflection. Okay, but there are two ways of achieving the TUF. One is the prism based and other one is the objective based. Here you can see the difference between the epifluorescence, confocal and TUF image. So in epifluorescence, you can see so much of background light, or I would say out of focus light. Okay. Then in confocal, the out of focus light is reduced, but still you can structures which are even in the deeper of the cell. Okay. But in the turf, you can only see the structures associated with the cover slip or the slide. Okay, so only the cover slip associated or the slide associated structures are illuminated, so that you cannot see anything within the cell. Okay, and that's why this turf is the best way to study the membrane bound. On this slide, you can see the comparison between the turf microscopy and confocal microscopy. Although we are going to discuss more about the confocal microscopy in the one of the next class, but you can just see how they are different. Okay, so in terms of the lateral resolution, the turf provide more or less the same lateral resolution like the confocal, so there is no gain of resolution here. But in terms of the axial resolution, turf provides better axial resolution. Axial resolution means resolution in Z. Okay, so if you take the Z stack, so as I mentioned that the turf microscopy cannot be used for taking the Z stack. It only gives you the information very close to the cover slip. Okay, it cannot give you international information throughout the cell. So that's why it provides better axial resolution of 50 to 100 nanometer, whereas in confocal microscopy, the axial resolution is 500 to 1000 nanometer, but confocal can take the Z-stack information of the cell, whereas turf microscopy cannot provide you the entire Z-stack information for the cell. Okay. Now, which kind of detector is used in turf microscopy? So generally CCD camera or the SCMOS, these days mostly the SCMOS camera are used, whereas the confocal microscopy, photomultiplier tubes are used as a detector. And here the detector sensitivity is 40 to 90% because of the SCMOS camera. And here because of the PMT, the detection sensitivity is around 10 to 25%. And as turf microscopy only illuminate the cell which is in close association with the cover slip. Okay, so it creates low phototoxicity, whereas confocal microscopy create medium to high phototoxicity because it can illuminate the entire cell volume. And imaging speed is fast because it has to image only one focal plane, whereas imaging speed is quite slow because in confocal microscopy it goes for line by line scanning that we'll discuss in the next class. Okay, and cost wise also this upgrading your microscope to turf illumination is quite cheaper compared to the confocal illumination. Okay. So here you can see a variety of applications of turf. In which kind of experiments you should use turf microscope? Because as you know that the turf microscopy provides you the information only for the cell structures which are very close to the membrane uh, or the proteins which are associated with the membranes. So this is the best technique for studying the secretory granules or vesicle trafficking, endocytosis, exocytosis, extracellular matrix structure and assembly, lipid and membrane protein biophysics, single molecule protein motion and dynamics, cell adhesion, migration, motility and mechanics, membrane trafficking dynamics, calcium sparks and ion channels, membrane and cytoskeleton remodeling, surface and interfacial chemistry, signal transduction, viral assembly and dynamics, and super resolution imaging. So you can see most of the applications are somehow membrane related or something which is happening at the membrane of the cell, not something which is happening in the nucleus. Okay. So let us move on to the next type of illumination that is known as highly inclined and laminated optical sheet illumination or the high-low illumination. Okay. So high-low illumination is same as the turf illumination. However, the incident angle is lower than the critical angle. Okay. So let me go back to this previous diagram. So here I explained that in turf, so this is the critical angle which is shown by the gray line. Okay, and for the turf, your incident angle, the blue line, is greater than the critical angle. Okay, for high-low illumination, this blue line is on this side of the gray line. For high-low illumination, your your angle of the illumination light should be lesser than the critical angle. Okay, and why is this so? That we'll discuss now. So see, in this diagram you can see for hydro illumination, your light will enter from this end, and sorry, for turf illumination the light will enter from this end. It will be totally internally reflected in the objective lens, and the light will come out from this side, okay, because it's a total internal reflection. Whereas for high-low, the light will enter from this point, and a thin sheet of light will pass through the sample like this. So here the light will not be internally reflected, but a thin sheet of light passes through the sample like this. Okay, so if I show you this diagram again that we already discussed, that how the light path is managed for the turf illumination. So just one step before turf illumination, you see this is a high-low illumination. So here the angle of the incident light is slightly lower than the critical angle, and that's why a thin sheet of light passes through the cell, okay, instead of the total internal reflection. So you can see that this is a cell or the specimen, and a thin sheet of light passes through the sample, okay. So this allows a thin sheet of light roughly around 3 micrometer to pass through the cell, okay, and this approach is widely used for single molecule imaging as it provides better signal to noise ratio for single molecules. So mostly for single molecule in live cells, this approach is used because it will only illuminate a few molecules in the cell and avoid the out of focus light from the molecules which are above or below the focal plane. So coming to this slide, and here you can see the comparison between the epifluorescence and high-low illumination. So in epifluorescence, this is your sample, and this is your focal plane, and the light enters straight away from the sample and it illuminates the entire cell, okay. And because of that, you will see this kind of an image. Only the molecules which are in the focal plane, you can see them perfectly focused, but the other molecules which are above or below the focal plane, they will provide out of focus light, and that will create noise in your image. Whereas in high-low illumination, a thin sheet of light passes through the cell nucleus, and because of that, you can only see the fluorescence from 3 molecules, and the other molecules, they will not get illuminated, and because of that they will not provide any out of focus light, okay. So using this high illumination, you cannot see all the molecules present in the nucleus, but what you can see is how these 3 molecules uh, behave inside the nucleus, whether they are dynamic. So suppose you want to study the dynamic of these 3 molecules, then the high illumination is best because you can see only 3 molecules and you can see that whether they are dynamic or not, okay. So coming to the next illumination strategy, that is light sheet fluorescence microscopy, LSFM. Okay. So this method is used for imaging thick samples such as tissue explants, three-dimensional cell culture, embryos, and entire organism such as C. elegans, zebrafish, etc. So this illumination strategy is generally used with the thick samples, the thick tissue samples, or for imaging entire animal like entire organism like C. elegans or the zebrafish. And in light sheet illumination, you can see here a thin sheet of light passes through the cell exactly at the focal plane. So only these many molecules are illuminated, and those illuminated molecules, the fluorescence from the illuminated molecules, get captured by the objective lens. This is the best approach for 3D imaging with low photo bleaching and low phototoxicity. Why low photo bleaching and low phototoxicity? Because you are illuminating this plane and you are collecting the information from this plane. Then you can illuminate the next plane, you can collect the information from that plane. You illuminate the other plane and you can collect the information from that plane. So it provides low bleaching, low photo bleaching and low phototoxicity. This is not an ideal method for imaging small organisms such as bacteria and yeast. So never try using light sheet microscopy for imaging bacteria or the yeast cells. You will never get good images using this approach. Okay, only with the thick tissue samples you should use this method. So now I will show you a few movies on the YouTube which shows the light sheet imaging of different organisms like C. elegans, zebrafish, and just see the beauty of this imaging. Okay, so I'll just show you these 3 videos one by one. So here you can see the light sheet microscopy of a zebrafish in vivo in the live cells. Okay, you see. So here you can see a zebrafish embryo multi-view fluorescence imaging. Here you can see zebrafish embryonic development. So you can just appreciate the power of this light sheet imaging so that you can image the entire organism like C. elegans zebrafish in 3 dimension, in even in 4 dimensions and 5 dimensions also. Okay, so you can do the Z-stack imaging using this light sheet microscopy. Only thing is every time your this light sheet will also move up and down along with your illumination plane. So how this light sheet microscopy works? So here you can see the optics alignment for the light sheet microscope. So if you see the light sheet microscope, you will find that there are two objective lenses perpendicular to each other. Okay. So for light sheet, see, for other type of microscopy, you just need one objective at a time. Okay. But for light sheet microscopy, you need two objective lenses. So the first objective is known as the illumination objective, which is generally of low numerical aperture. And the other one is the detection objective, which is generally of a high numerical aperture. So the purpose of illumination objective is to just pass a thin sheet of light through the sample at perpendicular to the detection objective. And the detection objective collects the fluorescent light emitted from this sample. Okay, so here the function is separate. See, generally in other type of microscopy, the illumination light also passes through the objective lens. It illuminates the sample, and the objective lens collects the fluorescent light. Okay, whereas here in light sheet microscopy, the illumination objective is separate. So light source is attached to the illumination objective, and it just illuminates the sample, but it will not collect the fluorescent light emitted by this sample. But that fluorescent light emitted by the sample, that will be collected by this detection objective. Okay, and see, sample is placed exactly at the center of both these objectives so that one can illuminate and the other can collect the fluorescent signal from this sample. So the sample alignment and the optics alignment is very critical in this light sheet fluorescence microscope. So here's some more details about the light sheet fluorescence microscopy. So with standard epifluorescence microscopy, the cone of illumination and detection is stretched in the axial Z direction, unnecessarily exciting significant out of focus fluorescence and degrading the quality of in focus signal. So here you can see the comparison. Left side is the light sheet illumination and right side is the epifluorescence illumination. So you see in the epifluorescence illumination, the entire specimen is illuminated, whereas in light sheet illumination, a thin sheet of light passes through the specimen. So if you see the photo damage, so see here only a thin sheet kind of photo damage you can see, whereas under the epifluorescence illumination, almost like 50% of the sample is photo bleached or photo damaged. Okay, so using this light sheet, you can reduce the photo bleaching and phototoxicity. Light sheet illumination provides better signal to noise with minimal sample exposure and high frame rates and long-term imaging. Okay. Axial confinement of the illumination results in dramatically reduced photo bleaching and phototoxicity, allowing long-term imaging over many days and even weeks. So as we saw the zebrafish embryonic development, right? So this process takes a few days. So you need to image that sample for few days. So obviously you need to reduce the photo bleaching and phototoxicity. So only with the light sheet microscopy it is possible to reduce phototoxicity and photo bleaching to so that your fluorescence will remain for at least few days. Okay, if you try this experiment with the epifluorescence microscope, then your fluorescence will be bleached in just within first few hours on the first day itself, and also it will create a lot of phototoxicity so that your embryo will die before it completes the development. Okay, and the disadvantages of this method is difficult to implement, difficult alignment process, and non-standard sample preparation protocol. So as you know, the sample should be perfectly placed perpendicular to. So here the sample is not there on the slide, but there is a special sample holder in which the sample should be held exactly at the this position so that it should be focused by both the objective lens. So this is a very tricky alignment process and it's difficult to align. Okay, and it is difficult to implement because it needs a specialized microscope because two objectives should be parallel, should be perpendicular to each other. Okay, and the non-standard sample preparation protocol. So it is little tricky. You need some expertise to work with this technique. It's not for the beginners. Okay, you need some expertise for sample preparation and imaging using light sheet microscopy. So coming to the two photon microscopy or the multi-photon microscopy. So when to use multi-photon microscopy? So multi-photon microscopy is also good for the thick samples from 200 micrometer to several millimeter, some tissue samples or some small organisms. It is also good for the live samples and it is excellent for imaging cellular details inside the live animals. Okay, so let us see how this two photon microscopy work and why the name is two photon. Okay, so whatever the other type of microscopy that we studied till now, they are all considered as a single photon excitation. Why single photon? Because for illuminating any dye, we give the light of, let's say, if you want to see GFP, you illuminate with the 488 nanometer. So single photon will excite the electrons from ground state to the higher orbit. And when those electrons goes back to the ground state, then it will emit the wavelength of the light of a higher wavelength, okay, which is known as the fluorescence. But in two photon, instead of a single photon, you will use two photon of a longer wavelength. So let's say if you want to see the GFP protein, so instead of using the 488 nanometer light, which is having more energy, you will use the light of a reduced energy, that is 976 nanometer light, okay, which is in the far red region or the infrared region. Okay, so you will use this infrared light. Okay, and you will use two photon so that when the two photon will illuminate the same point in the sample, at that time they can provide enough energy to excite the electrons from the ground state to the higher orbit, and those electrons will come back to the ground state by emitting the wavelength of a light of a higher wavelength. Okay, so this is known as the two photon excitation. So in two photon excitation, both photons of half the energy must arrive nearly simultaneously within less than 1 femtosecond time. So fluorescence intensity depends on the square of the excitation intensity. Okay, 1 femtosecond is 10 to the power minus second for 15 seconds. So here is another diagram to explain the same concept. So let's say you want to excite electrons from the ground state to the higher orbit. So there are three ways. This is a single photon excitation in which you will use the light of 360 nanometer, which has the higher energy. So with a single photon can excite the electrons from the ground state to the higher orbit. The other ways you can use the two photon excitation, in which you can use the light of 720 nanometer, so half the energy than this 360 nanometer light, and these two photons should arrive at the same position at the same time within 1 femtosecond so that they can impart the same energy to excite the electrons from the ground state to the higher orbit. And this is a three photon excitation. So if I use the wavelength of 1080 nanometer, then three photons should arrive at the same position at the same time within 1 femtosecond to provide the same energy so that the electrons can excited from the ground state to the higher orbit. And when all these electrons comes back to the come back to the ground state, they emit the light of a higher wavelength, that is 460 nanometer. Okay, so this is the concept behind two photon or multi-photon illumination. So most of the time, generally the two photon illumination is used, but there are several cases where the multi-photon, like three photon excitation, is also used. Okay. So what limits tissue penetration depth? Now let's say this technique is good if you have to image a thick tissue. Okay. Now what will happen if you use a single photon excitation? Just like the regular epifluorescence mode, if you use to image a thick tissue sample, then what will happen? It will illuminate the larger area, and also the absorption and scattering of light will take place because of the thickness of the tissue. Okay. Whereas in two photon excitation, we are using the near infrared light for the excitation that reduces absorption and scattering, and we are illuminating such a small area. Because why such a small area? Because this is the probability, this is the area where the probability of reaching two photons simultaneously to illuminate the to excite the electrons from the ground state to the higher orbit. So the small green area that is shown here, that is the area where two electrons will come simultaneously within 1 femtosecond to impart the energy which is enough to excite the electrons from the ground state to the higher orbit. So that's why in two photon illumination, only a small area is illuminated. Okay. So here you can see this is a one photon illumination. In this cuvette, some red colored dye solution is placed. And if you see the one photon illumination, you can see a slit kind of thing, right? So it illuminates this area. But if you see the two photon illumination here, can you see a tiny dot here? So it will illuminate only such a tiny spot in the sample. So if this is a tissue sample, it will only illuminate this tiny spot, and it will give you the fluorescence, and you will capture the fluorescence coming from this tiny spot. Now for two photon illumination, you need a specialized kind of a laser. The regular laser that we use for confocal microscopy or the TUF microscopy, that is not enough for two photon illumination, because emission intensity depends on the square of the excitation intensity. So you must have the very high excitation intensity, and those high excitation intensity you cannot get from the regular lasers. So our regular lasers are known as the continuous wavelength laser. So this is a typical continuous wavelength laser that we generally use for the confocal microscopy or the TUF microscopy, okay, in which the same wavelength is continuously emitted from the laser. But for this two photon microscopy, you need pulsed laser to get the high power but normal average power. Okay, so in pulse laser, you see the illumination is by pulse. Okay, it's not a continuous illumination. And uh, the name of the laser, this kind of lasers are generally known as the TI sapphire laser for two photon microscopy. And here are some physical parameters of this laser. The pulse width is 80 femtoseconds. The repetition rate is 80 lakhs per second. Time between pulses 12.5 nanosecond. 80 femtosecond pulse every 12.5 nanoseconds. So all these are just to show that how fast this laser works. Okay, everything is in femtosecond and nanosecond. Okay, the laser is only on for 1 by 1,50,000 of the times, and 1 watt average power results in 1,50,000 watt peak power, and this is tunable for the wavelength 700 to 1000 nanometer. So excitation wavelength is tunable from 700 to 1000 nanometer, and cost is generally USD 100,000 to 200,000 US dollars, or 1 crore INR. Okay, so this is a TI sapphire laser that is generally being used for the two photon microscopy. Now here is one interesting thing about the two photon microscopy. You see, this is a two photon excitation spectra. So you can see all different dyes, all four different colors of dyes. If you have, let's say, Alexa Fluor 350, 488, 568, and 594 nanometer dye, four different dyes. you do not need four different lasers for the illumination. Okay, see all the dyes under two photon excitation illumination thing, they can all excite at this wavelength. So if you have the laser which provides the wavelength of around 750 nanometer or 730 nanometer, then you can see most of the dyes, all the dyes gives you fluorescence. So you just need one laser to illuminate all the dyes at 750 nanometer or so. Okay, so this is broadness of spectrum, means one wavelength can excite multiple dyes. In general, all commonly used fluorescent proteins, CFP, GFP, YFP, RFP, and the dyes can be excited in two photon mode. Two photon excitation spectra are often considerably broader, making it more difficult to excite fluorophores selectively by switching the excitation wavelength. Okay, so in two photon, it is not possible that, let's say, if you just want to see the DAPI staining, you just illuminate the dye at 350 nanometer. Okay, or you can just eliminate the DAPI dye and not the others, because all dyes will get illuminated at this 750 nanometer wavelength, and that's why only one laser, that is the TI sapphire laser, is there in the two photon microscope, which can illuminate all four dyes. Okay, but now how to separate these colors? Okay, so for that purpose, two photon optics are simple and allow multiple channels to be collected simultaneously. So the different filters are placed in the emission light path. So you can see that this is the objective lens and this is 720 nanometer short pass filter. So the illumination light from the TI sapphire laser is roughly around to 720 or below that. So that light illuminates the sample. It will illuminate all the four colors, and all the four color fluorescent light will be collected by this objective lens. And then there is a one 750 SP. SP stands for short pass. Okay, so this short pass filter is there. So 750 uh, uh, nanometer short pass filter means it will allow all the wavelengths which are shorter than 750 nanometer. So our 495, 525, 565, 655, all this wavelength will pass from this filter, but the wavelength higher than 750 nanometer, they will not pass from this filter. And then there are dichroic mirrors are placed in the light path, and they will separate these colors individually. So this is a dichroic mirror which will reflect 475 nanometer, but it will pass all the longer wavelength. Then this dichroic mirror will reflect 510 nanometer and uh, transmit all the wavelengths higher than 510 nanometer. This dichroic mirror will reflect 542 nanometer light and transmit all the higher wavelength light. And this dichroic mirror will reflect 607 nanometer light and transmit all the higher wavelength light. And this is the last detector which can collect all the light at 675 nanometer. So this is how the four or five different colored fluorescence can be collected in the emission light path. Okay, using different dichroic and different filter sets. So in two photon microscopy, the excitation only one laser works. Okay, so that is 720 nanometer wavelength laser. It can illuminate all four dyes, but in the emission light path, there are multiple filters and dichroic cap placed to collect the signal individually in different channels. Here you can see one image of the two photon excitation microscopy of mouse intestine, and you can see the actin in red color, the cell nuclei are in green color, and the mucus of goblet cells in blue color. And this image has been obtained at 80 nanometer using TI sapphire laser. Okay, so with the same laser you can illuminate all these three dyes and you can collect the fluorescence in different channels. And again, in this two photon illumination also you can take the Z-stack images. Okay, so this is how the two photon microscopy works. So with this, let us end today's class, and our next class we will discuss about the super resolution microscopy, that is structured illumination microscopy, straight microscopy, then storm and palm microscopy. So there are 3 or 4 variants of the super resolution microscopy that we will discuss in the next class. Okay, thank you.